Hello dear friends and colleagues. I am Anuradha Chowdhary. I am Director Professor at Vallabhbhai Patel Chest Institute, University of Delhi, India. I work in the Medical Mycology Unit, which is also our National Reference Laboratory for antimicrobial resistance and fungal pathogens. Today I am here to discuss about the species identification as recommended in the Global Guidelines for Candida diseases. And here is the global guideline for the diagnosis and management of Candidiasis. It is a huge initiative of the ECMM in cooperation with International Society of Human and Animal Mycology, ESHAM, and also with the American Society of Microbiology. And I am here as one of the contributors of these guidelines. And this guidelines was recently published in Lancet Infectious Disease 2025. We have multiple small videos discussing all the essential aspects of this guideline, and today my job is to discuss the species identification of the Candida species as recommended in these guidelines. So once we come to the species identification, why it is important? Why it is important to identify correctly the Candida species? It is important because early identification helps in detecting the outbreaks, and also it is very important to implement infection prevention and control measures. Not only that, it's also important that we identify the Candida species accurately for surveillance studies and also to monitor antifungal resistance trends. And with that, I will now come to the steps which are used in the routine microbiology laboratory for identification of Candida species. So we all are familiar with Severos dextrose agar, which is the preferred isolation medium for Candida species as compared to other several medias which we use like blood agar, McConkey agar, primarily for bacteria. For Candida species, with Severos dextrose agar is the preferred isolation medium. Now we come to the other media, and which is a chromogenic agar media, which is a differential medium which provides different colors to different but some selective Candida species, and is routinely used in the microbiology laboratories. So this chromogenic agar media, it provides presumptive identification to the species level for some species, and this is especially important in mixed yeast infections, and it accounts, for example, the mixed yeast infections account for 2 to 3% of candidemia cases, and they are particularly concerning in patients after solid organ transplantation and surgery. And to select those mixed yeast infection, usage of chromogenic agar media is recommended. And it's also in some species, this medium can also be used for identification of clinically significant isolates, but those species has to be confirmed by other methods. And in our guidelines, chromogenic agar media is strongly recommended for the detection of mixed yeast infections, and it is moderately recommended for presumptive identifications, and those presumptive identifications has to be confirmed by the definitive test, which I'm going to discuss shortly. So we all are aware that standard mycological media or biochemical tests, they differentiate species based on the morphological characteristics. However, we all know that different species can share this similar morphological characteristics. So one of the example, if I give you a positive germ tube test, which is routinely used in some basic laboratories to differentiate albicans, typically identify Candida albicans, but we know that some strains also produce, or other than albican species, also produce these germ tubes. For example, Candida dubliensis also produce germ tubes. So just by identifying these morphological features, we cannot give the identification of the Candida species. and therefore these media are not recommended in routine microbiology labs for confirmed identification of Candida species. So in the present guidelines, biochemical methods are recommended with moderate strength for identification in settings, specifically in the laboratories where MALDI-TOF or sequencing are not available. So therefore, now we go to the MALDI-TOF, which in these guidelines is strongly recommended to provide species level identification of the culture isolates. And I'm going to discuss that in my subsequent slides. So for sequencing of the internal transcript spacer region or ribosomal D1, D2 region is generally considered to be the gold standard for fungal pathogens. To identify the species, for confirming these species, ITS and even D2 region sequencing is considered to be very important. However, we all know that sequencing facilities are not available in all the laboratories. Further, the species identification using sequencing is time consuming and expensive. However, wherever these facilities are available, sequencing is considered to be very important for species identification, and especially for the isolates which are new, which are not available in the database of MALDI-TOF or the commercial identification system. So the sequencing is strongly recommended, particularly in specialized laboratories. And also, if we come to the other tests like peptide nucleic acid fluorescent in situ hybridization, PNA FISH in short, PNA FISH is used for identification of Candida species primarily from positive blood cultures. But the unfortunate is that to date, commercially available kits have been discontinued. So practically the test is not available for usage. And also we are aware of several commercial PCR kits which are used for detection of Candida species from clinical samples and are available and have commercially available, and these kits have demonstrated good sensitivity and specificity targeting individual species, and therefore be used for identification of Candida species to the species level, provided that species is included in the kit panel. So finally, this is how the overall, I'm going to show you the recommendation panel in the guidelines. And if you see this panel, you will see different colors: green, yellow and pink. So green recommends suggests strong recommendation, yellow shows moderate recommendation, and orange or pink shows marginal recommendations. And this is how the species identification flow chart goes. And here identification, again I will emphasize that identification to the species level is strongly recommended if treatment is being considered, also to guide empiric management and to detect outbreaks, and also again I would like to emphasize for surveillance study. And as discussed in the previous slide, MALDI-TOF is strongly recommended to provide species level identification. And also, if you look at the chromogenic media, chromogenic agars, it shows green color, as these are strongly recommended for the detection of mixed yeast infections and moderately recommended for presumptive identification. We are aware of the biochemical tests and automated systems, and I discussed about the lacunae of biochemical tests. So biochemical methods are recommended with moderate strength for identification in settings where MALDI-TOF or, as discussed, sequencing are not available. And when we come to the PCR and we talk about the PCR, directly from the blood. So in the guidelines, PCR directly from blood is recommended with moderate strength because they only detect some Candida species. So therefore, the recommendation is with moderate strength. And if you look at the fish, PNA fish from positive blood cultures is recommended with marginal support. Only as with marginal support, and only as commercial kits are currently available. So finally, I would like to conclude that molecular identification of culture isolates by Molotov is strongly recommended to provide species level identification. Sequencing from culture isolate is strongly recommended, particularly in specialized laboratories and when identification could not be obtained by biochemical methods, or when the species is new and is not available in the database of Molotov. So with that, I hope this session was informative for you, and for details I will again refer to you. Please go to the Global Guideline Candidiasis Disease, Lance Infectious Disease 2025 and go through the document, and I hope you will you enjoyed this session and you enjoyed the reading the document. Thank you very much for your attention.